Hello viewers. So after the discussion of replication of DNA and experimental proof of replication of DNA, now today I am here to explain you what is the mechanism of DNA replication. What is the mechanism? You know that this DNA replication is a very accurate process, because if in the replication of DNA, even if there is a minor mistake, it may cause a large mutation, a very big mutation. And the process of replication of DNA, it is a costly process also, because a lot of energy is required in this process. So the replication of DNA, it starts from a specific point. The point from where replication of DNA starts, that point is called origin of replication, that is ORI. In prokaryotes, there is only one ORI, origin of replication, but in eukaryotes, there may be more than one ORI. So even in a single DNA, the process of replication in eukaryotes can start at several points, at several places. So when the process of DNA replication starts, as I told you, it is a very complicated process and it requires more than dozen enzymes. The two strands of DNA which remain wind up, they start separating from each other. This process of separation of two strands of DNA, it starts from the point of ORI, origin of replication, the process of separation, and it is controlled by the enzyme helicase. So when the two strands of DNA are separated, then this DNA, it becomes a Y-shaped structure. This Y-shaped structure, and this Y-shaped structure of DNA, it is called replication fork. On the two separated strands, on the two unwind strands of DNA, now the process of replication begins. On the separated strand of DNA, the new nucleotides of DNA, they begin to add, and they will be joined up by the enzyme DNA polymerase. But before starting the process of addition of nucleotides of DNA, there is formation of a primer. What is this primer? Primer is a segment of nucleotides of RNA. Primer is a set of nucleotide of RNA, and the enzyme which controls the synthesis of RNA is primase enzyme. The process of replication always starts from 5' terminal to 3' terminal. So new strand of DNA always begin to form from 5 terminal to 3 terminal. So on this template strand, where the template polarity is 3, the new strand starting from 5 terminal, and first there is primer formation, and then the process of replication proceed from 5 terminal to 3 terminal. Whereas on the another strand, again there is first formation of primer, and then new strand of DNA begin to synthesize from 5 terminal to 3 terminal. When this process of replication is completed, then the next segment of DNA is unwinded. And when that next segment of DNA is unwinded, then on that newly exposed DNA, the process of replication continues. On this strand, where replication already started from 5 terminal, the process of replication continues because it is already from 5 to 3. But on opposite strand, where this replication started from 5 terminal to 3 terminal, but here on this newly exposed area, again there is primer formation, and again there is synthesis of new strand of DNA. After this completion, then the next segment of DNA will become unwind, and on that newly synthesized exposed DNA on one strand, the process of replication continuously taking place, because already it is from 5 terminal and it is proceeding towards 3 terminal. But on opposite strand, as here, one segment of DNA started, one segment of DNA is synthesized, then another segment of DNA is synthesized, and then after that, on this newly exposed DNA, again there is primer formation, and again from 5 to 3 there is synthesis of new segment of DNA. It means the DNA are having 2 strands. From the 2 strands, on one strand, the replication of DNA is continuously taking place. On which segment? Whose 3' terminal is towards the primer. This replication of DNA is called continuous replication, and this strand of DNA on which continuous replication is taking place, this strand of DNA is called leading strand. Now the another strand whose 5' terminal is there, where? Towards ORI. Here the DNA is synthesized in small pieces. This synthesis of DNA is called discontinuous replication. It is discontinuous replication. So you can compare what is continuous replication and what is discontinuous replication. And this is strand of DNA on which discontinuous replication occurs. This segment of DNA is called lagging strand. So again, you must be able to differentiate which one is leading strand and which one is lagging strand. And on lagging strand, the discontinuous replication is taking place in the form of small pieces. These small pieces of DNA synthesized during discontinuous replication, these pieces are called Okazaki fragments. These are Okazaki fragments. Later on, what will happen? The primers are the nucleotides of RNA. So these primer pieces will be removed, and at the place of primer, the DNA nucleotides will be added. And finally, all these Okazaki fragments will be joined together with the help of enzyme DNA ligase. So this process of DNA replication, we also know it that it is semi-conservative replication, because in the 2 DNA, one strand is old, one strand is new. One strand is old, one strand is new. So this is the process of DNA replication. If you like this my video, then please like it, share it, and please subscribe my channel. Thank you very much.